Morning everyone. This is GIAS Academy, Rampuram, Chennai. We are giving online and offline courses. Helpline is given here. CSIR NET, Life Science, Unit 5. This class is about gametogenesis, fertilization and early development. Let us see what is happening in the sexual reproduction in animals. So this consists of 3 steps. One is gametogenesis, second one is mating, and third one is fertilization. What is gametogenesis in the sense? Production of gametes. Production of gametes in the sense it is male as well as female gametes. Male is sperm, male gamete is called as sperm, and a female gamete is called as ovum. Second one is mating. What is mating? Getting gametes together. That is, the sperm as well as the ovum is getting together. So that is called as mating. And what is fertilization? Fertilization is fusion of male as well as female gametes. See, for example, the sperm is having its nucleus and the ovum is also having a nucleus. The number of chromosomes is n number. So this is from father and this is from mother. When these two fuse, it form a zygote. That is called as fertilization. So during fertilization, the fusion of male as well as female gametes takes place. So there is a fusion of nucleus, haploid nucleus from sperm as well as ovum takes place and it forms a zygote. This is called as zygote, and the number of chromosome present here is 2n. That is one set from father and one set from mother. So the sexual reproduction of animals consists of 3 steps: gametogenesis, mating and fertilization. Let us see here. This is the process or the steps which takes place during the spermatogenesis, and this is the oogenesis. Let us see first what is happening during spermatogenesis. So spermatogonia is a single cell. This is called as spermatogonium. This is a single cell, and here what is happening in the sense, the pairing of chromosomes takes place. Pairing in the sense the homologous chromosome, that is one set from mother and one set from father. These homologous chromosomes, they pair together. Okay, this reduction is called as, division is called as reduction division. So what happened here, 2 chromosomes comes this side, and that is one mother chromosome and one father chromosome. That is, one is from mother, one is from father, come to this cell, and the next 2 come to the next cell. So what is happening? The 4 chromosomes are divided into 2. Okay, these are the daughter cells. So this daughter cell receives 2 chromosomes and this daughter cell receives only 2 chromosomes. Okay, whereas the mother cell in the sense the spermatogonium consists of 4 chromosomes. So the reduction is taking place here. So this reduction division, this division is called as reduction division or meiosis. Okay, so this is first phase of the meiosis. Second phase of meiosis, what is happening here? This will replicate and goes to the 2 chromosomes. This replicate and form gives rise to daughter cells, and this cell gives rise to daughter cells. Each daughter cells receives 2 chromosomes. So the number of chromosomes is n. N number of chromosome is present here, and this is called as spermatid. These cells are called as spermatid, and this cell is called as primary spermatocyte. This one, after the reduction division, that cell is called as secondary spermatocyte. You have to carefully note all the points. So primary spermatocyte consists of 2n number of chromosome. Secondary spermatocyte consists of n number of chromosome. Okay. And if you come to the daughter cells, each one divides into 2. So at the end of meiosis, there are 4 daughter cells. At the end of mitosis, only 2 daughter. But at the end end of meiosis cell division, there are 4 daughter cells, and these daughter cells in male it is called as spermatids. After maturation, it is called as spermatozoa. So it possesses a tail, so it is ready to move from one place to another place. Okay, another important thing what you have to keep in mind is, see, 4 spermatids are forming, right? So, or the 4 spermatozoa are forming. So if you see this one, half of the spermatozoa possess X chromosome, and half in the sense 50% X chromosome and other 50% is Y chromosome. So this is about the spermatogenesis. Spermatogenesis in the sense production of germs, production of sperm from spermatogonium. So at the end of the meiotic division, there are 4 spermatozoa which is ready to move, and among this 4, 50% possess X chromosome, and the other 50% possess the Y chromosome. Okay. And one more important thing I would like to say is what is happening here in the sense, these chromosome, if you see, the chromosome at the time of cell division, see, for example, this is one chromosome, and this is another chromosome which is from mother. Okay, homologous chromosome. Just imagine, we have seen in the picture there are 4 chromosomes. So one set is from mother and another set is from father, right? So homologous in the sense similar type of chromosome. The human being possess 23 pairs of chromosome. How many pairs in the sense? 23 pairs, and 22 are allosomes, 22 are, and one pair is sex chromosome. It is sex chromosome. One pair is sex chromosome. If it is female, it is XX. If it is male, it is XY. Okay. And if you come across this one, at the time of cell division, this chromosome divides into 2 chromatids. 2 chromatids. Okay, here also the same thing takes place. Now the chromatids, they separate in the sense they look like this. Let us take this is one chromosome. Okay, only this part I am taking, and another, this is another chromosome. The chromatids, they move apart. Okay, so this is called as crossing over. What is happening here in the sense, see, let me dot this one. Let me dot this one. This is the chromatid of chromosome A. Okay, just name it. This is chromosome A and this is chromosome B. So what has happened at this point? There is a exchange of gene takes place. Exchange in the sense a part of the chromosome is detached and move along with the next chromosome. Okay, so this is what in the sense what happened at the detachment. I'll take some other color and show you. See, this detachment, this come and join here. Okay, join here, and this one will take this part. Okay, so part of the chromatid from B joined to the chromatid A, and part of the chromatid A is now come and joined to the B chromosome. Okay, so like this they change. So the part of the genes which is present here now come to this chromosome, and part of the genes which is present here, or part of the chromosome which is present here, it moved toward that chromosome. Okay, so this is why the genes are transferring, and there's a character change or any other changes from the mother and father, the child is possess some other character. Okay, the color change or the way of behavior will be changed because of this exchange of genes, okay, between the chromosomes. So now let us see this. This is I told you it is spermatogonia, these cells, and this is Sertoli cells, which gives nutrients to this spermatogonia. And if you come to this one, this is these are the spermatid, which is not ready for the fertilization, that will mature and they form the spermatozoa. Now it is ready, and this moves from one region to another region, which is this is highly motile. Let us see what is oogenesis. Oogenesis, in the sense, the formation of ovum is called as oogenesis, and 1 month per month only one ovum is produced. See, whereas here what we have seen here is 4 daughter cells, whereas here there is only 1 daughter cells produced, that too n number of chromosome. Okay, so what is happening to the other n number of chromosome? In the sense, let us see here, this is oogonium. From the oogonium, the primary oocytes form. This possesses 2n number of chromosome, and here the first division, that is first meiosis, is a reduction division. So the n number is reduced to 2n number is reduced to n number. So this cell, that is secondary oocyte, possesses 2 chromosome, and the next part is called as first polar body. Okay, first polar body. And if you come here, the next division is the meiotic division 2. So here the secondary oocyte is transformed into otid as well as one polar body, second polar body. This is, so how many polar bodies are forming? In the sense, only 2, first polar as well as second polar. First division, first polar body, and second division, second polar body is forming. So two chromosomes are moving there and 2 chromosomes are here, and the cytoplasmic differentiation is very good here, and here it is very poor. So now how many cells are ready for fertilization? In the sense, it is only 1. Okay, so this ootid is now transformed into ovum. So the ovum consists of a number of chromosome, and also it is only 1, whereas here it is 4. In the sense, here only 1 egg is produced. Okay, coming to this one, this is fertilization. How the fertilization takes place? In the sense, the sperm pierces the membrane of the ovum, and it releases its nucleus. This nucleus combined with the egg nucleus, and zygote forms. Okay, this is called as fertilization. This is the structure of ovum, and what you are seeing here is this membrane is called a zona pellucida, and these are the follicle cells. First it has to pierce the follicle cell, then it has to penetrate the zona pellucida, and you can see here this is the first polar body. Okay, this is sperm nucleus and cortical granules. These are very, very important. After the penetration of this sperm, this cortical granules will be released, and it joins with this zona pellucida and form a thick layer, and this layer never allow the other sperms to enter into the egg. Okay, if you see in plants what is happening here, this is the male reproductive part, and this is female reproductive part. This part from here to here, it is female reproductive part, and this is the ovary. The ovules are arranged here. So what is happening here? In the sense, from the anther, the pollen grains are falling on the stigma. From the stigma, the pollen grains forms a tube. See, for example, this is, let us take, this is the stigma. I just enlarged and shown to you. And if the pollen grains fallen here, in the sense, it makes a tubule-like structure. It's a tube-like structure, minute tube, and the nucleus, only the nucleus from the pollen grain enter here. Okay, slowly from top to bottom it enters, and it reaches the ovule, and then their fertilization occur. So this nucleus with the ovular nucleus, the two nucleus combine and form the zygote. You can see here, this is the egg nucleus, and now after fusion the zygote forms. These are the endosperm nucleus. Endosperm is very very important in plant because which nourish the young plants, and this is the seed coat and this is the embryo. So if the condition is favorable, if the seed falls on a favorable conditional soil, then from the seed a seedling arises. Okay, so this is what happens in plants. See here, it is very given, very clearly it is given here. What is this is meiosis 1, and the first prophase. What is happening in the sense, the nuclear membrane starts disappearing and the centrioles move towards the opposite direction and spindle fiber formation takes place. And during metaphase, the chromosomes are come to the middle region of the cell, and the spindle fibers formed, and the nuclear membrane completely disappeared, and they arranged at the center and ready for the, and the spindle fiber starts to contract. And here you can see very clearly in the anaphase what is happening. See here, the homologous chromosomes are arranged, whereas here the homologous chromosomes are taken. So 4 chromosomes are reduced into 2. So 2 daughter cells are formed at the end of this reduction division. And you can see the mother cell possesses 4, in the sense the daughter cells, 2 daughter cells, they received only 2. So this is 2n number of chromosome now reduced to n number of chromosome. Okay. Now this is meiosis 1, this is meiosis 2. What is happening? The 2 daughter cells, you can see 2 daughter cells here. These 2 daughter cells, the nuclear membrane started disappearing, and the spindle formation and centriole move towards the opposite side. And then again during metaphase, the chromatids, the chromosomes are arranged at the center, and the chromosomes are divided into chromatids. Now during anaphase, they are pulled towards the opposite pole, and as a result, how many cells are formed? In the sense, there are 2 cells from each, in the sense these 2 are divide and form 2 cells. So totally it is 4 cells, and each cell possesses only n number of chromosome. Okay, this is spermatogenesis. Okay, and whereas in oogenesis only 1 egg will be produced. One is polar body, first polar body and second polar body. So only 1 ovum is ready for fertilization. You can see here the sperm is entering, and this part is of the this part of the sperm is called as acrosome, which is cone in shape, and this acrosome possesses hydrolytic enzymes. These enzymes are very very essential to digest the membrane. Which membrane? Zona pellucida, and it penetrate very easily. So you can see clearly this part is digested, and now the sperm entered little bit inside, and these are the cortical these are the protein receptors. So this is these receptors are called as protein receptors. These receptors only the sperm come and bind with this. Okay, and after digesting this one, it enters into this into the egg. There you can see the cortical granules. These granules, what it does, in the sense, they fuse with this membrane, zona pellucida, and as well as the vitelline membrane, which become very thicker and never allow the other sperm to enter into the egg. You can see the sperm movement. I will show you how the sperm is moving from one area to another area. You can see here numerous, that is million numbers of sperms are released inside the female body, and it moves towards the egg. What you are seeing here is the egg, and the sperms are moving and finding the receptors, the protein receptor. If it find the receptor, the fastest one, the first, and it is releasing the digestive enzyme, and that digests the wall of the egg, and now the homologous chromosomes are coming together and they are pairing. Now they form the zygote. This is a single cell zygote. Now divides into, it's ready to divide now. It divided into 2, then into 4, then into 8, then it is 16, and now it forms the blastula and it is moving towards the uterus. The fertilization takes place in the fallopian tube only. So the egg present when it comes from the ovary, is while it is moving towards the uterus, at that time in the in the fallopian tube, the fertilization takes place. And one more important point what you are going to see is the acrosomal process is coated with a membrane bound protein. That protein is called as bind-in. Okay, that will be different kind of bind-in molecules in different species. You can see this one very clearly. The binding of the sperm. See, when you take, this is the egg. This is the egg, just imagine, and this is the wall of the egg. Okay, see, this is which region in the sense, this is jelly coat. Okay, jelly coat which covers the egg. You can find a jelly coat here. Okay, just above the egg, and you can see this is the vitelline envelope. And this is plasma membrane, egg plasma membrane. This is vitelline envelope. Just above the vitelline envelope, the bind-in are there. Okay, the sperm binding receptors are bind-in receptors. So the sperm which comes and attach with the receptor. Okay, the attach with the receptor. So now what happen? It releases its enzyme and penetrate into the vitelline envelope, and it is releasing. Here you can see it is releasing. At that time, what is happening? When it is penetrate, in the sense, immediately the cortical granules, they fuse with this vitelline envelope and make this. Okay, this cortical granules absorbs water, and the vitelline envelope swells and become very thick, and it never allows the other sperms to enter inside. You can see this is the sperm nucleus. So the sperm nucleus is released here. Now it is ready to fuse with the egg nucleus and the fertilization occur. Okay, the zygote is the result. And you can see there is a slow block to the polyspermy in sea urchin that is mediated by phosphatidyl inositol biphosphate. Okay, and also one more is there, that is IP3, which diffuses the endoplasmic reticulum, where it opens the calcium channel at the time of fertilization. And one more important thing what you are going to see is female oyster. What you are seeing here is the female oyster. There are two types of fertilization. One is internal fertilization. Okay, internal fertilization. What is internal fertilization? In the sense which takes place, fertilization takes place inside the body. Okay, inside the body, in the sense it is called as internal fertilization. Okay, if it is outside the body, the fertilization takes place outside the body, then it is called as external fertilization. Okay, the fertilization is called as external fertilization. You can see here. So let us take one example. Let us take the oyster. Okay, this is oyster. The female oyster, for example, may produce 100 million eggs at a time. Okay, 100 million eggs in a year. per year it lays 100 million eggs, the oyster female, and the number of sperms which is produced in male oyster is astronomical. Okay, it is numerous sperms are produced. What you are seeing here is the eggs of oyster. It is in magnified way which is given. Okay, this is magnified way it is given, the egg of the oyster. You can see how this oyster is releasing eggs in the water, in the sea water. You can see now. I'll show that. So you can see here this oyster is releasing a cloud like solution. You are seeing, right? It is pumping out into the water. This cloud possesses this possesses numerous eggs. Okay, this is female which is releasing the egg. Next one, I'll show you how the sperm are released in the sense. Just a minute. You can see here, these are the male oyster. They are releasing the sperm. They are releasing the sperm into the water. That is, millions and millions of sperms are released inside the water. So in the water only the fertilization takes place. Okay, the eggs are released and the sperms are also releasing in the water. So the fertilization is external fertilization. Now, what is amniote egg in the sense? Some of the birds, okay, birds as well as reptiles, they lay the eggs, and the embryo which develops inside the egg. Okay, egg in the sense the outside it is covered with a hard shell, and inside the food material, that is, which is called as yolk, which is a preserved food material for the developing embryo. So that egg is called as amniote egg. And some are oviparous in the sense the egg laying animals are called as oviparous, and viviparous in the sense they give birth. Viviparous animals give birth the young ones. They give birth young ones, so they are called as viviparous. Oviviviparous in the sense they lay the egg. The egg is present inside the body, and that the embryo develops. That is called as oviviparous. And one more important thing what we are going to see is, you can see here how the fertilization takes place in the sense. See, if the male and female differentiation, if you see, always the ovum is X. Okay, ovum is always possessing the sex chromosome only X. Okay, ovum is X. If it is sperm, if you take, there are two types of sperm. One possesses X chromosome and the other possesses Y chromosome. Okay, so if the ovum is fertilized, with the X sperm, then the baby is a female baby. The female baby possesses 2 X chromosomes. Okay, if the ovum fertilized by Y sperm in the sense, then the baby is a boy baby. Okay, the boy baby possesses XY. Okay, XY sex chromosomes. So that is what here. And one more thing, if you see, the symmetry of the cytoplasm is not same. It differs from one region to another region. Okay, this is very important for the development of different layers, that is mesoderm, endoderm and ectoderm, and there is different Development, that is morphogenesis, okay, differentiation, everything takes place only because of the cytoplasmic differentiation. We see here, you can see very clearly how this differentiation are present in the egg. See, these are the frog egg, okay, frog eggs. The eggs are black in color, which is present inside the jelly, okay, so jelly coat it is. And you can see here, this is the animal pole. Only in the animal pole the nucleus present, and this is vegetative pole, and this is unfertilized egg, okay, this is not zygote, this is egg, okay. And see, you can see in the animal pole region is very dark, and it is highly concentrated pigmented animal hemisphere, this is. And here the cytoplasmic is filled with the yolk, okay, yolk. And this is vegetative hemisphere, this is animal hemisphere, and this is vegetative hemisphere. Here dark pigments are present here in more in number, whereas here it is yolk is present in the vegetative part, okay. So this is a single cell, but you can see in the cytoplasm there is so many variations are we are seeing here, okay. So these variations are responsible for the determination, differentiation, and morphogenesis while developing into the embryo, okay. And one more thing, what we are going to see is the sperm enter only the animal pole region. The sperm enter only the animal pole region. So the cortical cytoplasm rotate towards the site of the sperm entry. This rotation reveals a band. This rotation reveals a band, and which form, okay, band which is pigmented cytoplasm on the opposite side of the sperm entry. So this side there is a band formation. This is called as gray crescent. Gray crescent, okay. This is just opposite to the sperm entry area. And what we see is the sperm entry area forms a belly region of the embryo, and this crescent will become the dorsal region of the embryo, okay. So I hope you could have understand this class very nicely, and all the other links are given in the description box, so go through that and get through the CSIR very easily. Thank you so much. Share this video to your friends, those who are preparing for the CSIR NET, okay. The address as well as the our email ID and our website is given here, helpline also here, so you can contact our GIAS Academy and you can enroll it. Thank you so much.